So we will have a quiz today. It'll be the first of, I guess it'll be the extra credit quiz for you guys. The first of the extra credit quizzes for the online people. And it's going to include content that I go over today. So it won't be elephants on acid at all. It'll just be genetics and a little bit of DNA that we go over. Any questions so far? So you guys, you're all done with lab, right? I mean, you submitted your reports by now, hopefully, and you've taken your lab practical, so you guys are done with lab. Next Tuesday we will have lab. Okay. So we left off last time talking about genetics and Mendel, and we kind of went through some of the exceptions to Mendelian genetics. And there's really one more phenomenon that we're going to talk about that is problematic with respect to Mendelian genetics. And that is a process called DNA methylation. And it's a part of an emerging field within the study of genetics. And this is a really, really interesting and cutting-edge sort of field. It's called epigenetics. And what it suggests is that there's some environmental characteristics outside of Mendel's laws that affect whether or not, or impact whether or not, an allele will be expressed. So if we remove all of these kinds of environmental and cellular effects, in the presence of like a heterozygous individual that has both a dominant and a recessive allele, we know, right, that that dominant allele is going to be expressed. So in the context of flower color, if a flower has both a dominant capital P purple allele and a recessive white allele, that flower is going to be purple. What epigenetics suggests, though, is that some things can happen on a cellular level, which may be a product of environment or may be a product of microcellular environment, where, for whatever reason, that dominant allele is deactivated, and so the recessive allele is expressed. Now, that happens because of some cellular kinds of things that we do to our DNA. Specifically, we go through a process called methylation, or DNA methylation. And that can happen one of two ways, and I'll talk about both of those in just a second. But in essence, what happens is the mechanism is a methyl group, which is just a chemical group, is applied to different regions of the DNA. So like an extra chemical is added on to the DNA, and what that causes the DNA to do is to bind up really, really tightly, as if it was like a mitotic chromosome. So that that region of DNA isn't accessible to cellular machinery for the expression of that particular gene region. Now, if it happens on the DNA, typically methylation occurs at CG dinucleotide sequences. So we know, right, in DNA that A binds to T and C binds to G. For whatever reason, that purine-pyrimidine combination of C and G are more amenable to DNA methylation. And so at those dinucleotide sequences, you get extra methyl groups applied, and that DNA spins up really, really tightly, and that region becomes inaccessible to cellular machinery. A really good example of this is the X chromosome. So you women, you all have 2 X chromosomes, but at any given point, only a single X chromosome is active in each of your cells. So functionally, in each cell, you only have a single X chromosome. Now that ought to sound a little odd, and it does sound a little odd, but for the following reason. If you think back to when we were talking about meiosis, and homologous pairing, I mentioned the observation, right, that some sorts of cognitive disorders are more common among males than they are among females. Do you remember that observation? And do you remember the explanation for that? Yeah, and the X chromosome codes for lots of cognitive processes. So if males inherit a faulty X chromosome, we don't have homologous genes on the Y chromosome to compensate for those genetic inadequacies, right? which means that those cognitive disorders are more likely to be expressed among males than among females. But functionally, females, you're the same, right? Because you only have a single X chromosome that's active within your genome at any point in time. So how do we resolve those two rather disparate and confusing observations? That is, that you only have one X chromosome active, but it's lots less likely for you to express cognitive disorders. What are your thoughts on that? Yeah, what do you think?
You're saying there's the homologous chromosome in X, there's two X chromosomes for females.
Yep.
So wouldn't, when one's not working, our body's pretty good at figuring out what's not working, and so it would activate that second mutant chromosome to compensate for the lack in the
And something similar happens to that, but instead of it being an intentional process, it's a random process. So we have trillions of cells throughout our bodies, right? And if the cell, each cell is randomly choosing which of those two X chromosomes to deactivate through DNA methylation, the result is approximately 50% of your X chromosomes will have the correct gene on it, and 50% will have a faulty gene on it. And that's sufficient enough to compensate for any genetic inadequacies on that deficient X chromosome. So as a product of some random process, some random selection, deactivation, you ladies are covered. Genetically, you're good. Okay, does that make sense? Okay. So in addition to actually methylating the DNA, we can also methylate those proteins around which DNAs bind or wind. And those are called histones, right? And so we can hypermethylate histones, and it's going to deactivate large regions of DNA. What's really interesting about this, you guys, is there is a correlation between DNA methylation and cancer. So there is a region of our genome called the dark genome, and it's called the dark genome because we don't really know exactly what it does. What we have noticed, though, is that there's a correlation between methylation of the dark genome and the probability of cancer. So within a given cell, if the dark genome is hypermethylated, what that means is that cell is a lot more likely to develop into a cancerous cell than something that isn't methylated. Okay, so the reason we're talking about epigenetics again is because this can be a violation of Mendel's hypotheses, right? What this can do is, regardless of whether an allele is dominant or recessive, we can deactivate it through methylation. Now, I should also say that this process of DNA methylation is how we get gene expression in some cells that isn't expressed in other cells. Because all of our cells have exactly the same DNA in them, right? But we don't want eye genes being expressed on our skin, right? Because that we're not seeing with our skin. Neither do we want like hand genes expressed in our eyes, right? That would be disgusting. So what we're able to do is we're able to deactivate those genes in specific regions of our body through this process of methylation as well. Okay? All right. So the question is, what really is the unit of inheritance? Is it a gene or is it a chromosome? That is, what is it that's actually determining patterns of phenotypes? And ultimately, right, we know that it is a gene that determines an allele, but we're receiving those genes in clusters, in packages, and those packages are the chromosomes. And so arguably, we could state that the chromosome is the actual unit of inheritance because it's on chromosomes that genes are located. Now, we know that there can be recombination in genes as a product of synapsis and crossing over. And so there's considerable variation, and indeed, even though multiple genes are located on a single chromosome, they can behave as though they're being inherited independently of one another. So really what this is supposed to be is a segue into a conversation about what's called gene linkage. When you think about this, you guys, what Mendel said was each character is inherited independently, right? Just because you're inheriting one character doesn't mean that you have to inherit any other characters. They're all independent of one another. And if that was absolutely true, what that would mean is, for each one of our genes, it has to be physically and functionally independent, as if each gene has its own unique chromosome. But we know that's not true, right? Within the human genome, there are like 22,000 different genes, or 22 to 24,000 different genes, lots and lots of genes. And how many chromosomes do we have? 46, 23 homologous pairs, right? So across those 23 homologous pairs, there are 24,000 different genes that are distributed. So what that means is, some genes are more likely to be inherited together, those that are existing on the same chromosome and relatively close together on the same chromosome are more likely to be inherited together than genes that are located on completely different chromosomes or far apart on the same chromosome. So this is the phenomenon of linkage. What we can do is, based on patterns of inheritance, we can determine how close two genes are located on the same chromosome. Now, as it turned out, Mendel was really fortuitous in the kinds of characters that he was looking at. So he looked at, you'll recall, flower color, seed color, pod color, seed shape, pod shape, and stature. 6 different characters. 5 of those characters are located on different chromosomes, but 2 of them ended up being located on the same chromosome, although they were located very far apart on the same chromosome. So we can look at the distribution of genes across a chromosome. So let's define a couple of chromosomes, and we're going to use post-anaphase chromosomes, right, that are just have one sister chromatid. So we have a homologous chromosome from mom, and we have that same homologue from dad, like so. Now what we could say is, on those chromosomes, there are located 3 genes, and we'll call them a gene for eye color. So this blue gene is eye color. We'll say there is a green gene, and this green gene is for hair color. And then one more genetic character. Do you guys know what hitchhiker's thumb is? Yeah, it's rad. So most people's thumbs look like this, right? But that's about as far back as you can bend them. But with hitchhiker's thumb, it goes boom, all the way back like that. So that's hitchhiker's thumb. What's crazy is hitchhiker's thumb is a product of a single gene locus that follows Mendelian genetics. So individuals with this kind of a rad thumb are homozygous recessive. Individuals that have a normal thumb, unfortunately, are dominant. Either they're homozygous dominant or they're heterozygous, right? So we're going to say this last gene is hitchhiker's thumb. So this is mom up here. This is dad. What we're going to say is, and we're not going to like define what these alleles actually mean. We're going to say that mom, for eye color, has a dominant allele, and dad has a recessive allele. And that's going to be true for all of these genes. Mom has a dominant allele, and dad has the recessive allele. And then we're going to make this one D. Mom has a dominant allele, dad has recessive allele. Now we know, right, that during meiosis, during prophase 1, synapsis occurs and crossing over occurs, right? And we might get crossing over that looks like this, right? Where this part of dad's leg flops over on mom's leg, and those two chromosome regions are exchanged. So that at the end, after crossing over has occurred, we get this distribution. Mom still has a dominant one here, but she inherited dad's recessive and dad's recessive. Likewise, dad still has a dominant one here, but he inherited mom's dominant and mom's dominant. Still really dad's chromosome, right? He just took on some of mom's genes. That's called a recombinant chromosome because it has DNA from two different sources on it. So what we can do is we can look at rates of recombination. Now, with rates of recombination, what happens, right, is the higher the rate of recombination, the more frequent crossing over is occurring. And if crossing over is truly a random process, which it is, what that means is the farther apart two gene regions are, the more likely it is that recombination is going to occur, or the crossing over is going to occur. So, if we had the following data, and I'm going to define, see if I can do this in my head, I'm going to define 3 other gene regions, and you guys are going to tell me how they're located on a chromosome. Okay, so we have 3 genes. We have A, B, and D. And we're going to say that the recombination rate between A and B is 62%. We're going to say between B and D, it is 24%. And then we're going to say